Now, I'm here today, I'm wearing two hats. One as the CTO of Oxford Nanopore, and I'm going to talk about that technology in the context of DNA being the next silicon, a very catchy tagline. But also, we have our own startup activity. We've been spinning out little companies for a couple of years, a little company called Metacore. I'm going to try and put today's technology talk in context of Metacore. Now, as Javier said, Oxford Nanopore has been around for about 10 years, and we've been commercializing or seeking to develop and then commercialize nanopore DNA sequencing for about 8 years, and with about 250 million pounds of input capital. Now, we haven't spent all of that yet, thankfully, but as you can see, what I'm about to talk about cost a lot of money. And whilst it superficially looks very simple, I think the real trick has been to make something that is quite difficult look very simple. So let's see if we can get the next slide up. So let's talk about DNA. And I don't know your backgrounds, but I'll try and put this into context. So obviously, every living thing contains DNA. It still surprises me that when people talk about DNA sequencing in food or genetic modification, the general public are quite queasy about DNA. I think many of them don't realize that everything, every beef burger, every vegetable themselves is full of DNA. Every plant, everything. And even now, I am shedding DNA onto this stage. My cells are dropping off, the bacteria on my skin are dropping off. And the same applies to you. It's all over the place. Everything you touch, you leave DNA behind you. And DNA moves. Obviously, when people move and when things move around, the DNA moves with them. And they leave traces. And those traces can be detected in principle. Now, what is DNA sequencing? Well, DNA sequencing is what it means is we're going to decode a molecule. The molecule itself is a double helical, very long polymer that is in every cell. But when we talk about sequencing it, we talk about converting it into ostensibly 4 letters. And those 4 letters are the code. They're the code from which we can then extract and analyze much more information about genetics and biology. And the molecule is a polymeric nature. The 4 letters are sequential. And living things have radically different amounts of DNA in their cells. So, for example, each of my cells contains 3 to 6 billion base pairs of DNA, and there may be 13 trillion cells in my body. A bacteria might contain 4 million bases of DNA. But there might be more bacteria in me and on me than my own cells, to put that into context. So obviously, a very different scale in terms of decoding these things and trying to understand what's going on. From a virus or a bacterium that might be a few thousand or a few million letters to a human being that can be billions and billions. So why is it important? Well, from DNA, you can identify what something is. You can say, what is it? You can also say, where does it come from, in terms of traceability. DNA changes. I think this has been one of the interesting things that's happened over the past 5 years, is that we know that every cell mutates. It's the basis of the generation of cancer. Bacteria mutate and grow. Antibiotic resistance in particular is caused by evolution. DNA gets modified. We may hear later about how there are more than 4 bases, actually. It turns out there are quite a few more than 4 bases, and they're probably significant biologically. So by reading that DNA, we can ask whether an organism is or has changed. Very important question. Then obviously from that, we can ask deeper questions. For example, is it diseased? Is it normal? Is it dangerous? And so forth. So just from scanning one molecule, we can extract an enormous amount of dynamic information. Some of it historical, some of it predictive. Now, why is it important? Well, let's think about DNA being the new silicon. I think we've all lived through the internet era. I remember it starting in my mind in the late 80s when you had to dial up, but now you can send billions of bytes of information down a wire. You can download things and watch them. Well, in your body, in your blood vessels, every cell that's moving through there is packed full of DNA. The cells in your body are dying and they're shedding DNA, and that DNA is being transported around your body. The cells in your body communicate with each other through little packets of DNA. When viruses infect you, they're in the blood. So in a way, if you were to look at a blood vessel, every second you have billions and billions of bytes of dynamic information flowing through that blood vessel, all of which recording and reflecting what is happening or what has happened in your body. But the same applies to a water pipe. They're very often full of bacteria and viruses, the water that comes through your tap. The same applies even to things like a subway system where people move and flow. And in fact, people have sequenced microbes on subway systems, and they've been able to identify people, and so forth. So by analogy, the DNA that flows through these systems is rather like information flowing down a wire. Now, the trick is, can we tap into that wire, and can we decode what's going on and treat it like internet information? Now, even one drop of blood, and when we started talking about this 5 years ago, this was a slightly out there idea. But now there isn't one company in this space that isn't working to decode what is happening inside one drop of blood. One drop of blood contains thousands of cells, and those cells can be your immune system, where your immune system has seen and reacted to something in your body, whether it's cancer or an infection, and they've recorded it in them, and they've often amplified it too. You have free-floating DNA from cells which have died. You have, particularly easy to detect in a way, is aberrant DNA that's come from cancer cells which have died. Even the bacteria on your teeth will shed DNA into your bloodstream. So one drop of DNA contains trillions and trillions of bytes of dynamic and useful information. And there's an enormous race on now from large drug companies through to technology companies to figure out how to inventory, decode, and understand what is being recorded in that one drop being a sample from that pipe of information. And again, when we were talking about this 5 years ago, this is really, apart from what you're about to hear later in terms of editing DNA and synthesizing DNA, this is one of the hottest areas of biological research right now. And it's called liquid biopsy, and I wish I'd thought of that phrase. It's really catchy. Mine was far worse. So for example, what's in there? Well, this is old news now. This slide is a couple of years old. But it was discovered some years ago that, for example, a pregnant woman, her fetus will shed its DNA into her bloodstream, and the DNA of the fetus can be fully sequenced from the mother's blood. And this has become a routine test in some parts of the world now, where you can actually sequence the genome of an unborn child and detect genetic diseases before the child is born. Very powerful. There was a paper about 6 months ago, or perhaps longer, where they were screening pregnant women for genetic defects in unborn children, and they were accidentally finding cancer DNA. And they were pre-diagnosing cancer in the pregnant woman before the tumor appeared, before a lump appeared, effectively. That's how powerful this is. This is just within humans. Of course, the same applies to animals. In principle, the same applies to all kinds of systems. It's been shown that you can detect cancer DNA in a person with cancer, obviously, even at quite low levels, lower than the size of a pea in terms of the lump. And that sensitivity is getting better every day. And it's even been shown that when you cut the cancer out, still the best treatment for cancer is surgical removal. You can see a concomitant drop in the amount of DNA, cancer DNA. And the amount of cancer DNA that's left is indicative, often, of how effective that surgery has been. So clearly these are quite important medical, clinical, and there's a whole world of them now. Again, when I first spoke about this 2 years ago, these were quite shocking. You can Google this stuff now. You will get hundreds and hundreds of similar papers. So this is important. And just to show you how aberrant DNA can be, on the right here you've got a well-known cancer genome, and on the left you've got a normal genome, shown at the chromosome level, zoomed out. And you can see just how aberrant that cancer is. It is amazing these cells survive, in fact, given how messed up cancer DNA can be. And in a way that's good for us because it's actually quite an easy thing to detect because it's so messed up versus normal. And probably most of the money right now in terms of liquid biopsy is going into detecting and understanding cancer DNA in central labs. And I say in central labs because if you were to sequence these things probably now, you'd be looking at something like this. These are the kind of machines that people use. The theme here is DNA the next silicon, and in a way, these are the supercomputers, or the mainframes of DNA sequencing. Typically, boxes can cost millions of dollars, or certainly around $500,000 to $10 million. They are quite infrastructure intensive. People need to be experts to run them. Quite a large number of labs now where there are arrays and arrays of these huge machines churning away, doing hundreds of human genomes per week. Now, that's quite a triumph, really, and I used to work on the technology on the bottom right here. This is really state-of-the-art. But our technology isn't like that, because what we've done is we have come up with a way to read DNA directly using tiny holes. A nanopore is a small hole. That's all it means. It's a hole that's 1.2 nanometers across. Think about that. It's actually not much wider than the DNA molecule itself. And what we do is we've constructed a custom silicon chip that can measure electrical current at a very sensitive level. That's on the bottom right of this slide. And on there's an array, so there's a 2D grid of little holes. And these are protein holes that we've grown and cloned out of bacteria. And they're balanced on soap bubbles. And each soap bubble is about the width of a human hair. And we've constructed this chip such that you can make it and send it through the post. It turns up and it works just fine. And what's shown here is the system. It's not really this big, by the way. People haven't asked me, is that to scale? No, it's much smaller than shown here. But here's a big version. And what you're seeing is a double-stranded piece of DNA zipping through a nanopore, zipping through a tiny hole, and generating a characteristic electrical signal. And using fancy state-of-the-art neural networks, we can decode that signal into those 4 bases. And within seconds, we can sequence a genome or an organism. And this has been around, I said this has taken 8 years, arguably, most of the past 4 years. This has been on the market now for 2 years. And this is the device that we've been selling. It's called a MinION. And it is a USB device. Contrast with the previous instruments. It's a USB device, no external power supply. It plugs into your laptop. You put sample in, and it decodes the DNA by forcing it through those little holes, balanced on the top of those little soap bubbles. And it's very fast, and it actually generates quite a large amount of data. If you've got one right now, I'm not selling, by the way, today, but if you've got one right now, you could probably generate about 10 gigabases. That's 10 billion bases of information from that, which is about 3 human genomes worth, approximately. Now, we are in the process of developing another device. I brought one with me. This is called Submarines. And all we've done is make it even smaller, and even faster, and even cheaper. And we've made a version that now plugs into a mobile phone with a much cheaper consumable chip. And the idea then is to make this ultra-portable in a format that can be run by anybody, anywhere. Our goal being to enable anybody to be able to sequence anything, anywhere. This is the next generation. This will be available next year. So you can see that by interfacing biology directly with electronics, we can do some really powerful things. And whilst it's early days, it's only 2 years in, we think there will be a shift from the kind of centralized mainframe decoding of DNA to a more mobile, more desktop version that is more ubiquitous, that is accessible to anybody and not run by experts. Now, where could these devices go? Well, the same sensor can be put anywhere. It can be put in manufacturing processes, it can be put in food production lines, it can be used on the farm, it can be used at home. Anywhere where there's dynamic biology, you'll be able to quickly scan and inventory that biology, and then with the help of analytics on the internet, you'll be able to understand what's going on. This is what we see as the future, and we call this the Internet of Living Things. That's the tagline that we came up with. And it's not a fantasy, it's sort of happening right now. A big example would probably be the recent tracking of the Ebola outbreak in Africa, where our devices were used, and other people's devices too. But probably the most immediate and the most local was we had local scientists running MinIONs and decoding the DNA of the Ebola outbreak. We've recently had people looking at infectious diseases. A key thing there is antibiotic resistance, obviously, is a major problem. And people now take them everywhere. People take them to the North Pole, they take them onto beaches, and they sequence in situ. So it's kind of half happening. And there's even one in space. We took NASA, did most of the work on this. They took a MinION, they took some flow cells, and they sent it up into space. And the picture on the top left there is the astronaut who leads the project running the sequencer in zero gravity. And they have a particular objective. Obviously, if you want to go to Mars, you want to find life on other planets, you're going to at some point need a DNA sequencer that isn't the size of a truck. Equally, it was also sent into submarines, and people are sampling from coral reefs and then measuring things in the submarine. So it can go anywhere, and that means anywhere. There will be a future where it's embedded in doors and devices, and your phone security might not be a fingerprint, it might be a DNA sensor. In principle, these things can go anywhere. And so the team who did most of the Ebola work, they collaborated with other groups, they pooled the data, and the Rambo lab came up with this lovely animation where the evolution of the Ebola outbreak was tracked over time and by geography. So we have a time course, and we have location stamps on all the data. And what they're able to do then, what the black lines are showing, are routes of transmission of Ebola through West Africa. And this led to some real actionable information, because it was quite clear that some of the people who were infected were traveling quite long distances on known routes and then seeding infections in other provinces. I should say the colors here are the strains, and the black lines are the movement. And so the authorities were able to act to close off transmission routes and hopefully dampen down the spread of that disease. And again, in future, this sort of dynamic outbreak monitoring, where you can in real time take action, intercession reaction, that might become a routine application of this kind of technology. But of course, it could apply to beef burgers that contain horse meat. It could apply to people on the subway. It could apply to any application where biology is moving and needs to be tracked. Another big one recently, which is going to be quite important, using this device, people can now look for infections in urine and detect the presence of antibiotic resistance. This is a big problem. Ideally, you want to have the test done while the patient is in the clinic, while you're waiting. And these infectious diseases, especially UTIs, they're very pernicious, people die of them. There's a real problem now with resistance to antibiotics. It's a global problem. In some countries, it's rife. And at least being able to let the patient leave with the correct antibiotic can have quite a large impact. And whether to prescribe that antibiotic or not, also a very large impact. So this will now get better and better, quicker and quicker. So we think this is so important that Oxford Nanopore makes the devices. It's Intel. And we have a spin-out called Metacore, which is going to place these devices, is going to market them, and most importantly, it will provide real-time, cloud-based analytics to enable people to track and decode what is happening when these devices measure some dynamic biological process in the environment. And that is ticking along very nicely, and I'm hoping we'll be showing some impressive deals over the coming weeks or months, where we can take this concept and really make it a concrete proposition that people can understand. My personal favorite is measuring yourself. Every month or every week, I want to, from either blood or spit, I want to see what's happening in my body. I think it is interesting. I think you will see infections in there before you get sick. I think you'll see cancers before you get sick. I think you can probably profile your immune system and really see what's going on in some detail. I think people can share these data, and they can collaborate as patient interest groups. I think it could be very transformative to go to your doctor with your information on your mobile phone, where you've got a time series that tells a doctor or an expert what has been happening over the past year, either prior to treatment or during treatment. I think what's called self-quantification could be a massive, massive application. So with that, I will stop, and that was the Internet of Living Things. And I'll thank the organizers for inviting us, and start with that. It's very nice to be here in Paris, and I hope you enjoy the rest of the talks. Thank you.